Welcome to OmicsBox, Bioinformatics Made Easy.
Okay, so as mentioned, I'm Marta Venegas. I'm a bioinformatician here at Bioban, and today I will give you a brief introduction to the single-cell cell type prediction, the current state, and challenges. So, since you are already here, I assume that you already know about single-cell RNA sequencing and that it aims to provide expression profiles of individual cells in counterpart of the typical bulk RNA-seq analysis, which allows a more granular analysis. And it has already proven to be really useful on deciphering the heterogeneity of complex tissues and has allowed researchers to build really complex and detailed cell atlases. And this information has been really useful on deciphering the cell type-specific biological processes that can vary between different diseases, conditions, or states. But despite its popularity, the single-cell RNA-seq analysis is still complex. So here we have some statistics from the single-cell RNA tools website, which currently tracks almost 2,000 tools in over 30 categories. And they are mostly written in R and Python. So this vast number of tools make it difficult to design a straightforward pipeline, and still this type of analysis is out of hand or for researchers which are not familiar with coding. But what does a typical single-cell RNA-seq pipeline looks like? So we will start with the quantification. We will quantify our reads to obtain the cell per gene expression values. Afterwards, we will do some quality checks and filtering to remove those low quality cells. And then we will start with commonly the first analysis steps, which is the clustering that tries to identify groups of cells with similar expression patterns, which already will give us some hints about the biology of our dataset. Then we will typically go to the cell type prediction to identify which cell types are in our dataset, so we can start asking biological questions to our data. For example, which are the differences in gene expression between cell types or within the same cell type in different conditions, or how are my cell types ordered in pseudotime, and so on and so forth. So, as we see here, the cell type prediction is a key and critical step of the analysis because the biological insights we will gain in the downstream analysis will depend on that cell type prediction and will be based on the cell types that we predict. So let's dive deeper into the cell type prediction. So, as I already mentioned, it consists of inferring the cell types of the cells present in our dataset based on some prior knowledge. So there are two main types of prediction algorithms: the marker-based algorithms that they use a list of a few marker genes that altogether identify a certain cell type, and the cells that have a high level of expression of those marker genes will be labeled with that cell type. On the other hand, the reference-based prediction, they use entire pre-annotated single-cell dataset as references, and there are two types. By correlation, in which they put our query dataset and the reference dataset in the same dimensional space, and the cell type is assigned based on proximity in that space. On the other hand, the classification methods compare the entire gene expression patterns of our cells and the cells in the reference and assign the label based on that gene expression similarity. And what are the challenges of these prediction methods? So with the reference-based prediction, it highly depends on the reference used. And ideally, this reference should contain all the cell types in our query dataset, and experimental design has a big impact in that. So here we have an example in which we have different samples in which the cells have been extracted using different techniques at the laboratory level, and we see that the abundances of the different cell types really vary in between experiments. And it won't be the same to use this as my reference than this experiment. It's not going to be that easy to find the erythroids, for example, using this reference than using this other one. On the other hand, finding a good reference is not easy nor quick because there are numerous databases and the reference comes with different file formats, and very importantly, we have to ensure that the gene IDs used in our dataset match the ones in the reference, and etc. And lastly, we are assuming that the annotation is accurate, but there are different curation qualities, and the decisions made by the researchers can introduce some biases here. So for all these reasons, it's advisable to use more than one references and compare the results, but as we see, sometimes it's not easy to find even one proper reference. Then, regarding the marker-based prediction algorithms, since they rely only on a list of few marker genes, they are more sensitive to technical factors that can arise during sample preparation or during sequencing. And moreover, the databases commonly are not consistent, so they usually give different lists of marker genes for the same cell types. And lastly, it has been seen that they have been outperformed by reference-based methods by many different benchmarking studies. So, without giving too many spoilers, since Ashwini will talk about this later, I would like to introduce the methodology or the knowledge base developed by Combinatics CellKV. So CellKV combines a robust cell type prediction algorithm with a manually curated and extensive knowledge base that includes thousands of publications. And in order to predict the cell types in our dataset, our query is compared against multiple references at a time. And the key advantages of this methodology is that first, you don't need to look for suitable references, so there's no need to worry for data format handling, and moreover, the knowledge base has standardized cell ontologies, as well as an integrated tool for gene identifier mapping, which also allows for cross-species annotation. And it has different levels of granularity, which also makes it available to identify really specific cell types. And finally, by leveraging the power of multiple references, it reduces the bias introduced by the experimental design, technical factor, and incorrect annotation. And the CellKV is part of the whole OmicsBox single-cell RNA-seq pipeline that covers from the quantification down to biological insights, so you can start right from your raw reads, and it supports both UMI-based like 10x and Drop-seq, or full-length like smarter single-cell RNA-seq technologies. And all the computation is performed in the cloud, so you don't really have to worry about the computational resources of your local computer. And it also includes an interactive UMAP TSNE Beware, in which you can explore your data and select the groups of cells that interest to you and perform custom annotations. And all of this, it can be accessed by a graphical interface. So there's no need for coding, no setup, no maintenance. Everything is simplified, so you just have to have fun analyzing your data. So let's start with the demonstration now. So here we have the OmicsBox platform, where the different analysis modules are located on the top left corner. In order to start with a single-cell analysis, we would go to transcriptomics and single-cell RNA-seq. We can start right from the raw sequencing reads and perform the quantification for both cell barcoded and full-length technologies, or we can load pre-existing count matrices into OmicsBox. Many different file formats are available and start our analysis from there. So in my previous webinar, I already covered the steps of quantification, QC, filtering, clustering, and even some downstream analysis and data exploration. So since the focus of this webinar is the cell type prediction, I will open some clustering results directly and move from there. Okay, so the main results table has two sections. The left part, we have the cells present on the dataset, their cluster labels, and some statistics assigned. And on the right part, we have the genes. And in the side panel, there's the different downstream analysis options available, as well as some result charts, and the UMAP TSNE viewer. So the projection can be changed here. And we can also change some colors. And the viewer is interactive in the sense that we can select cells with the lasso or the other tools, or by adding some categorical or gene expression rules. And we can add those selections to custom groups. Okay, but at this stage, we only have cluster labels, and we would like to see which cells are available in our dataset. So in order to perform the cell type prediction, we will go to Actions, Cell Type Prediction, and CellKB. So the wizard allows to configure the analysis just within a few clicks, and previous to run CellKB, we need the list of differentially expressed genes for each of the groups we want to annotate. And we can select the test here, as well as which group we want to annotate. In this case, it's Seurat clusters, but we can perform the annotation on any group present on the object metadata. To configure the analysis, the first thing is to select the input species, the species of our data, and the species we want to compare against. We can select a different one here, and we will do a cross-species annotation. Then the reference knowledge base can also be filtered out to ensure a more tailored annotation. And we can apply filters on the tissues, conditions, or cell type just by start typing and clicking on the option. Or if we press space, we can see all the available options. We can add as many as we want, and we can remove them or clear the entire filters. So once everything is set up, we just click on Run, and all the data and the analysis is performed in the cloud. We will receive the results after a while. And the main results of the cell type prediction are the labels, which are stored in the object, and we can visualize them in the UMAP viewer. We will obtain some broad and some granular annotations from CellKB, and we will also obtain a summary report to further inspect these results. So for every of the groups that have been annotated, we will have a more general and a more specific annotation, as well as the top candidate cell types with an associated score. So this score really is a relative measure, meaning that a high score means that this cell type is a better match than the other candidates. And if the score is similar, it means that these three candidates are more or less an equal match to our group. So in order to further inspect this prediction, we can click on the top hits button, and another summary will show up with the top 10 gene list that best match our group. So in this case, we see that the rank-based scores, which quantifies the similarity between the two gene lists, are fairly similar between the different candidates, but there are more matches to the pericycle cell type. That's why it has been the final annotation. On the other hand, here for cluster 3, we see that the root cap has a comparatively higher score than the rest of the cell types, and that's why it has been the final annotation. And we can go so forth, continue inspecting the results. Okay, so if we do have a good reference, we can also perform the cell type prediction with single-R. So this particular dataset consists of Arabidopsis root cells that have been treated or that have been grown in normal and high sucrose mediums. And the single cell library has been constructed with a drop-seq technique. And I was able to find in the single cell plant database a reference with the same growing conditions and also obtained with drop-seq, which makes a good candidate to perform, or a good reference to perform the prediction. And that's what I did with single-R. So let's see how the results look like. In order to configure the annotation with single-R, we first need to provide the reference, and we have to select which group of the cell metadata contains the cell types. And very importantly, we have to ensure that the gene IDs between our dataset and the reference dataset match. And we can load the reference into OmicsBox previously to see if they are compatible. And after some more adjustments, we will click on Run. And after a short processing time, we will obtain the results. But we already open them and display them side by side so we can compare. Okay. Let me paint it by the annotation. Okay. So in general terms, we see a high degree of agreement between the two annotations. For instance, the root endodermis here in brown has been applied to the same cells, as well as the root cortex label here in red. But some differences arise. For instance, here in the middle, we have some cells assigned to the root stel by single-R, whereas with CellKB, they have been assigned to the cambium and the pericycle, which are cell types that are inside the root stel, which is a more general term. So in fact, the CellKB annotation is more granular for this case. There's also a difference here with these cells that are labeled as root hair by single-R annotation, whereas CellKB distinguishes between the parent trichroblast cell type and the more mature root hair cell. Then on the other hand, on the left part of the UMAP, we have some cells that have been assigned to different subcell types of the root cap, but they are placed differently, or they are labeled differently on the two annotations. So since the root cap and the subcell types are a continuum, this part of these cells will require further inspections. But what happens when we don't really have a good reference? So I also performed the single R annotation, but using a reference where the cells have been induced with estradiol. But the sequencing library was also constructed with DropSeq, and we start to see more differences here. And there are some cell types that are still labeled the same. For instance, the root endoderm is here, but in the estradiol annotation, we see a group of cells here that have been mislabeled as root cortex. Root cortex. And for instance, we see that this cell type is spread along the UMAP, as well as the non-hair label here in purple. In fact, here on the bottom part of the UMAP, we see some cells that have been labeled as root hair, as with the previous annotation, but some of them have been labeled as non-hair cells. So in overall, we see that the annotation with this reference is a little bit noisier and less specific, with, of course, will impact in the downstream analysis and the biological insight that we will get by analyzing this annotation. So, as some concluding remarks, I think that these examples effectively show the importance of choosing a right reference to perform the annotation. And we've seen that with CellKV, we achieve reliable annotations, but without the need of looking for that reference in databases. And on the other hand, I hope that this walkthrough, the short walkthrough through the platform has demonstrated how easy and intuitive it is to perform cell annotation with OmicsBox. And I encourage you again to take a look into my previous webinar where I covered all the steps of the full single cell analysis pipeline.